Assalamu alaikum, dear students. In the previous lecture, we discussed tissue biochemistry and narrowed it down to the connective tissue, its definition, and its classification based on the nature of matrix. We also clarified an important concept that blood is special connective tissue where cells are functionally dominant. Now in this lecture, we will focus on the extracellular matrix, which is the most important biochemical component of solid and semi-solid connective tissue. So what is the extracellular matrix? The extracellular matrix, commonly abbreviated as ECM, refers to the non-cellular material present in space between connective tissue cells. This space is also known as extracellular or intercellular space. In many connective tissue, this space is quite prominent, and instead of being empty, it is filled with a complex network of macromolecules. These macromolecules collectively form the extracellular matrix. From a tissue biochemistry perspective, the ECM is not just a supporting substance. It is a biologically and biochemically active structure that directly influences the strength, flexibility, filtration, adhesion, and organization of connective tissues. Now, what are the sources of extracellular matrix components? An important point to be understood is that ECM components are not produced randomly. They are synthesized and secreted by connective tissue cells. In most connective tissues, the principal cells responsible for ECM synthesis is fibroblast. In specialized connective tissues, this role is taken over by chondroblast in cartilage, osteoblast in bone. Thus, the composition of the extracellular matrix varies depending on the type of connective tissue and the cells involved. The major function of the extracellular matrix include the provision of mechanical strength to tissue such as tendon and ligament. It also gives elasticity to tissues like skin and blood vessels. It acts as a filter, for example, in the kidney glomerulus. It also allows cell adhesions, helping cell attach, migrate, and communicate. Therefore, the ECM is not merely structural, it is functionally dynamic. Biochemical composition of the extracellular matrix include three major components, that is glycosaminoglycan and proteoglycans, fibrous proteins, and specialized extracellular matrix, also known as basal lamina. We will discuss now each of these components stepwise. So the first one is glucosaminoglycans. These are commonly known as GAGs. GAGs are long, unbranched polysaccharide chain. They are made up of repeating disaccharide units. Each disaccharide unit consists of one amino sugar that is either N-acetylglucosamine or N-acetylgalactosamine and one uronic acid. Because GAGs contain multiple carboxyl and sulfate groups, they are highly negative charged. This negative charge attracts water, giving the matrix its hydrated gel-like nature. This property is extremely important because it allows connective tissue to resist compression and absorb shock. There are 6 major classes of glucosaminoglycans. The first one is hyaluronic acid, chondroitin sulfate, heparan sulfate, keratan sulfate, heparin, and dermatan sulfate. These GAGs differ in their sugar composition, linkage type, and degree of sulfation, which explain their varied distribution and function in different tissues. So among these 6 groups, we will only discuss the hyaluronic acid, which is a unique glucosaminoglycan because it is not sulfated and does not attach directly to a core protein. It is composed of repeating units of glucuronic acid and N-acetyl glucosamine. Hyaluronic acid is a major component of synovial fluid where it acts as a lubricant, and it also contributes to the viscosity of connective tissue ground substances. Excess hyaluronic acid can be broken down by the enzyme hyaluronidase. The second is proteoglycan. So, in most connective tissue, glucose aminoglycans are attached to protein, forming complex known as proteoglycans. A proteoglycan consists of a core protein and multiple GAGs chain attached to it. The attachment occurs through a special link trisaccharide, which connect the GAGs to serine residue on the core protein. Proteoglycan give the matrix high viscosity, resistance to compression, and space filling properties. They play a key role in determining the physical characteristic of connective tissue. The third part of the connective tissue is the fibrous protein of the extracellular matrix, which provides structural and functional support. These proteins are divided into structural fibers and adhesive fibers. We will discuss these fibers in detail. The first one is the collagen fiber, which is the most abundant protein in the extracellular matrix of connective tissue. It is strong, fibrous, structural protein that provides tensile strength. The basic molecule unit of collagen is topocollagen, which assembles into fibrils and then into fibers. Different types of collagens are found in different tissues. Type 1, 2, 3, and 4 form fibrils, while type 4 collagen is non-fibrillar and is major component of the basal lamina. The second fiber is elastic fiber, which is made up of the protein elastin. These are elastic in nature, found in tissues like skin, lungs, and large blood vessels. It is hydrophobic, cross-linked protein that allows tissue to stretch and coil much like a rubber band. This elasticity is essential for normal physiological function. The extracellular matrix also contains adhesive glycoproteins, mainly fibronectin and laminin. Fibronectin helps cells attach to collagen, GAGs, and cell surface receptor, thereby organizing the extracellular matrix. Laminin is a major component of the basal lamina and play a crucial role in anchoring epithelial cells to underlying tissue. Basal lamina is another part of the connective tissue. These are specialized extracellular matrix. The basal lamina is a thin sheet-like specialized extracellular matrix found beneath epithelial cells and surrounding certain other cell types. It is mainly composed of type 4 collagen, laminin, heparan sulfate, proteoglycan. Functionally, the basal lamina acts as a selective filter, maintains cell polarity, facilitates cell migration. So, in this lecture, we discussed the extracellular matrix, its origin, functions, and biochemical components. We saw how the ECM play a central role in determining the structure and function of connective tissue, especially solid and semi-solid one. Thank you.